Let's look at calcium method of uptake. Calcium is absorbed as a divalent cation. It moves by bulk flow into the apoplast. Remember, it doesn't do well in the cytoplasm, but moves by bulk flow in the apoplast, which is in between the cells, which is often where it gets lodged and helps harden up things. It is absorbed in the tip of the growing xylem and not through cells with well-developed Casparian strips. Once you've got a well-developed Casparian strip, this will effectively block calcium absorption. When we look at this, basically what happens is if you've got the endodermis in what they call state 2 or 3, that is where the Casparian strip is well developed and you do not get any calcium influx. There may be passageways in between these where calcium may get through. Normally, calcium is going to be absorbed down by the root tip, where it is going to be able to move directly into the xylem in the central stele. It is allowed to go upwards as part of the fluids that are being transported. It doesn't get loaded in, it goes up in the xylem. The plasmodesmata are also impermeable to calcium ions. They don't pick up calcium and they don't allow it to move upwards. Calcium is only translocated in the xylem. When we're dealing with xylem is only moving towards the growing part. There is no mechanism by which calcium can be released at the growing point and move downward in the plant. It's only going to move towards the apices. You only get translocation in the xylem. This becomes very important when we look at what happens with calcium. As far as sufficiency goes, standard literature will tell you that it's between 1 and 5% depending on species and time of analysis. When we look at this, we're looking at somewhere between 1 and 50 milligrams calcium per gram of dry matter. It is not extremely concentrated compared to some of the other materials. Monocots need less calcium than dicots, and that's very important. The literature shows that on ryegrass, maximum growth rate of tissue had a concentration of 0.7 milligrams per gram dry mass, whereas maximum growth rate on tomato, the tissue concentration was 12.9 milligrams per gram dry mass. That is a difference of about 15 times. It is much more important in the growth of dicots than it is in monocots. This also could be very interesting. Monocots tend to have more C4 photosynthesis. If you've got C4 photosynthesis, if calcium gets into the cytosol, it's going to inhibit pepcarboxylase, which means C4 photosynthesis is not going to function. When we deal with algae, even though we consider algae to be plants, they don't have a really good developed cell wall. If they don't have a really good developed cell wall, they don't need a lot of calcium in the middle lamella. Therefore, in algae, calcium is actually a micronutrient. Calcium deficiency is seen in the newly formed leaves. It's not transferred out of areas where it's already been produced. Therefore, where you get the deficiency is the things that are starting to grow. Calcium deficiency reduces division of the meristem. If it reduces division of the meristem, you are not going to get branching. Deficiency, you get a reduction of growth, a weakening of tissue. You may also get a red-brown discoloration. That red-brown discoloration is often followed by necrosis. A shortage of calcium causes a general collapse of membrane and cell wall structure. Remember, it was important in the membranes to hold them together. It's important in the cell wall to hold that together. That will allow leakage of phenolic precursors into the cytoplasm. And if phenolic precursors in the cytoplasm are toxic, that can be quite problematic. When we look at it, deficiency is in young growing tissue and not in the old growing tissue. And it's very different from any of those where the ions are mobile within the plant. This shows you a plant with a calcium deficiency. The older parts of the plant seem to be all right. The newer parts are not expanding properly. You're not getting proper growth on it. And that's due to a calcium deficiency in that tissue. This shows you a localized calcium deficiency. This is called blossom end rot of tomato. This is often a very important disorder of tomato. It's due to a nutrient imbalance. It's a temporary nutrient imbalance, because as the tomatoes start to grow and they need a lot of calcium, if you don't have sufficient amounts of calcium in the soil, the part that's growing will start to suffer a deficiency. Once you start that, those tomatoes are never going to recover from that. That doesn't mean you can't use the tomato. You're going to have to cut that part off that's not functioning properly, but it's caused by a local deficiency of calcium. The way to suppress this is to make sure that you've got sufficient amounts of calcium. In some cases, they actually spray calcium on as a foliar spray on plants at stages like this to make sure that they've got enough calcium so that the blossom and rot does not occur. We have that type of deficiency as well. Luxury consumption. Calcium is often luxury consumed. If it's up to 5% in the plants, that's not really how much it needs. Calcium is formed into calcium oxalate. Calcium oxalate is common in most plant families. Up to 90% of the total calcium in individual plants can be found in calcium oxalate. These are little crystals. They're like little daggers on the inside of the cell. You can tell when a plant has calcium oxalate because you can actually feel that when you're eating the tissue. It's got these little spine-like things that seem to jump out and stick you. The formation of these crystals occurs in specialized cells, which are called crystal etioblasts. The calcium oxalate is osmotically inactive. These are crystals, so therefore it is in a form that is not water soluble. These crystals serve to lower the concentration of calcium in the apoplast of surrounding cells without affecting the osmotic balance of the tissue. Even though it's a divalent cation, you've taken it out of solution, so it's no longer in the ionic form. It's not going to affect the osmotic balance of that particular type of tissue. Toxicity. No reports of calcium toxicity have been documented. Even though calcium can do a lot of things in the plant, we never think of calcium as being a toxic type material.